this weekend, like, hey, do a project, just make sure you probably can have that and just send it, and I'll put it together, whatever. I still okay,
all right, so we're gonna start out with some questions for you. If I were to show you a picture of something that looked like this, you would say, Deborah, that looks like what? And the way you would know, because you would look for that thymine. Perfect, so deal done. That would be question number 1 correct. Now let's say I was pretending to be an enzyme and I landed on my strand of DNA and I was able to unwrap the helix and separate the strands. Who am I? So I have jumped on the DNA strands, I've unwound the helix, separated the strands. My name would be helicase. Perfect. So DNA unwound and separated by helicase. Now let's say I'm another enzyme. I hop on the DNA and I read T on the strand and I find an A out in the cytoplasm in a bacterial cell or in the nucleus of a eukaryotic cell. I see an A, I'll find T. I read C, I'm going to be looking for G. I read T, look for A, read A, find T, find C. Remember, these nucleotides are floating free. Read them, bring them up, bind them together. Does it look like I am creating a new polymer of DNA?
Yes.
And my name would be DNA polymerase. So DNA polymerase creates a polymer of DNA. And this is the process that's called what? Replication. You got it. So this would be the process of replication. Starting with the original strand and then replicating a new strand. The other thing I want to mention is, notice in DNA replication, you'll have one old strand and one new strand, one old and one new, and we call that form of replication semi-conservative. So semi-conservative because it has conserved one old and one new. Alright, so that's DNA replication done. Pretty easy. Any questions? Nope. Then let's move on to another thing that you can do with your DNA that actually happens more often than DNA replication. And start with your original DNA, double-stranded. Now I am pretending to be an enzyme. This enzyme can hook onto the DNA, and it will hook onto the DNA at the beginning of a gene. And there's a specific region on this gene that tells this enzyme where to start and where to end. Remember, in the beginning of a gene, and what do you think this region would be called? Let me maybe start with the end of the region of the gene. And it reminds me of an Arnold Schwarzenegger movie. Terminator. So Terminator sequence would be down at the end of the gene. beginning would be called the promoter region. So this enzyme binds to the DNA around that promoter region, and that signals this enzyme to begin its process of copying the DNA. The enzyme lands on the DNA, separates the strands, and now will read T, and it's going to find the complementary nucleotide A, read A, find U, read C, find G, read read T, find A, and you're getting the idea of this process. What's the name of this enzyme performing this process? And let's think about it. Does it look like I'm creating a polymer of sorts? And I'm creating a polymer of RNA. So my name would be RNA polymerase. Creates an RNA polymer. And this is the process of transcription, correct? Because remember, transcription is just a copy, however, it is a complementary copy of the DNA. But do you want to work with that original precious DNA, or do you just want to take a copy and then work on it? Want to protect that original DNA. So it's best to make a copy of it, and we'll make the copy in the form of RNA. So when RNA polymerase reaches the terminator site, that RNA transcript will now be released from the DNA, and the DNA will re-zip up. And now I just have a long strand of RNA. This RNA still needs to be decoded. And if you had this long strand, let's just say, do you see this sequence is identical here? AUG, AUC, AUG, AUC. So I'm going to take like the cooking show shortcut, say, pretend I've transcribed that DNA into the following sequence. And now this transcript is going to be decoded using the following tools that you see up here. So let's name what those little tools are here. Number one, what would you call this molecule here?
Messenger RNA.
This will be messenger RNA because it will needs to be decoded. Needs decoded. We call that decoding process the process of translation. And what are the ingredients that we need to have in that translation reaction? You may see this big red blob. What may that look like to you? Ribosome. So here will be your ribosome. It's like Pictionary all over again. And then you see these little yellow circles. You're thinking those look like amino acids. I'm not going to say the answer. And then this weird looking thing, number one, you'll note you see a sequence of 3, and what type of molecule does this tell you it is? RNA, since you see that U. What specific type of RNA is it? Your guess would be it's not a single strand like messenger RNA. but it's more in a clover leaf shape. So that will be your transfer RNA. And these will be some of the tools that we'll need to use. What's the transfer RNA molecule going to be transferring? Should not be a trick question. Amino acids, correct. So you might notice that these tRNA molecules need to be loaded with their amino acid. Can I just randomly, we'll just put this one together here, or is there a more methodical process that needs to take place? So there is a specific binding between the amino acid and the tRNA molecule, and you'll have to figure that out and help me. I used to have these labeled on the back. A couple of them I do. But let's use your genetic code that I had passed out to you yesterday. If you don't have yours with you, I do have some extras up here, so you can come up and grab some extra sheets. And if you want to share, I have 2 genetic codes per sheet, so feel free to tear it in half. All right. So a couple of confusing things about this molecule, the tRNA molecules. What would you call that set of 3 nucleotides in transfer RNA? Yes, that will be your anticodon. And that anticodon sequence is going to be complementary to what sequence? The codon sequence, and the codon sequence you'll find in messenger RNA. With these genetic codes, what you'll notice is these are all of the codons, correct? So if we need to figure out what the anticodon is, you're just going to look at the complement. So let's start with something like valine. If you look at valine, what are the possible codon sequences for valine? So valine codons. I will say on my chart I see 4 of them. Would you agree with that? Mm-hmm. Okay, so GUU, GUC, GUA, GUG. And if these are the codons, then I'm going to say the anticodons would be what? CAA, and then it kind of does take some thinking, huh? It is hard. It's to really concentrate a little bit. All right, so these are possible anticodon sequences for that amino acid valine. So if I have valine, I'm going to load up valine onto which of these 4 tRNA molecules? I see CAU as being the only one that matches the correct criteria. All right, so CAU, that anticodon is going to be loaded with valine. And then MET, that is the only amino acid I do want you to memorize. So MET will be the codon AUG, it's the start codon. And that means the anticodon for AUG is UOUAC. Correct. So I'll take my MET amino acid and load it onto UAC. Okay, now I just have 2 more tRNA molecules to load up. And I'll let you tell me which is which. Glutamine and isoleucine. Isoleucine, let's start with this one. Which transfer RNA molecule will I load up with isoleucine? GUC or UAG? So you have to look at quite a few of the codon combinations and anti-codon combinations, but I'm pretty certain you're going to find isoleucine loaded onto UAG. And then if you looked at the code or using your powers of deduction, you would say glutamine is going to be loaded at this transfer RNA molecule. All right, so we've loaded the transfer RNA molecules up with their corresponding amino acids. Now let's get the process of translation going. One thing I do want to point out is it's very common for an RNA transcript to have quite a bit of extra RNA at the beginning of the messenger RNA strand. So what will happen is the ribosome will start reading those first few nucleotides of the messenger RNA. But how does the ribosome know where to start translation? It's looking for the start codon. That was the only codon I want you to memorize. And that start codon, you already told me, is AUG. So when the ribosome reads UGA, nope, that's not it. Move to the next one, nope. Keep going, nope. And it's going to be searching for this start codon also using the help of that transfer RNA molecule. So you'll have that transfer RNA molecule binding. Is this the correct complementary match? Nope. Keep moving. Nope. Nope. Until finally you see the match take place. So the start codon is identified now. So now the ribosome starts translation, and it will then move to the next codon. And what is the transfer RNA anticodon it's looking for? UAG. Sometimes there will be a transfer RNA molecule that comes into the ribosome to try to match, but if it's not a complementary match, it will just come back out, and then it will try another one. And is there a match? Yes. So this will signal a match. Now the transfer RNA molecule will transfer its amino acid to the ribosome and the growing protein chain. The ribosome does have a few sites on it that we did not talk about in lecture. The A site or the acceptor site, the P, which is the peptide binding site or the bonding site, and the E site for exiting. For our class, you won't need to know A, P, E, but when you see it, it's just accepting that codon and the tRNA molecule. But when you have those two transfer RNA molecules in there with their amino acids, and they're both matches, now you will have that peptide bond made. Ribosome will move down to the next codon, and now this transfer RNA molecule pops out of the ribosome. Notice it has transferred its amino acid, so it's unloaded. And now what do you think is going to happen to that transfer RNA molecule? It's going to go get another MET. So pick up another amino acid to be used again, since it's just one of the translating tools. So now we have another possible match. Is this a match? Yep, so a peptide bond will be formed here. Moves down. That tRNA exits the ribosome. And we have our final tRNA molecule coming up. Peptide bond forming. Maybe. Ribosome moves down. Picks out that transfer RNA molecule. Gets to UAG, and you'll notice if you look at the genetic code, UAG codes for stop. And that's the signal to the ribosome that you're done translating. These transfer RNA molecules are finished, and you're left with your final protein, which is just your polymer of amino acids. And remember, it's not only important for the cell to know what needs to be produced as a protein, but there will also be a message to the cell on where to put that protein as well.
So are those amino acids directly bound into that RNA strand, or does that RNA strand go off and produce more?
To this RNA strand, no. So the only thing the amino acid was bound to
Or each other?
was originally the transfer RNA molecules. When the transfer RNA molecules transfers it to the ribosome, it will just be bound to its neighboring amino acid. So one of the questions I'll be asking will be, that is not correct. Of course, it has to be the last one. One of the questions I'll be asking is, what is the actual physical link between mRNA and the amino acid? Because no, they're not ever touching.
So it's that tRNA?
It will be the tRNA that serves as that link or the decoding tool between them. But you can see how all of these players in the translation reaction work together to translate the nucleic acid message into a protein, and that protein is usually going to form an enzyme or perhaps a structure that will be used somewhere in the cell. All right, so any questions on that? Nope. Then let's move on to mutations. And one thing I will always say is it's extremely important to know how things are supposed to work correctly, just like we've gone through, so that you can make predictions on what's going to happen if things go wrong. So mutations will be when things go wrong. Would you say you would think a mutation would be in the RNA or DNA? So for a mutation,
DNA.
Mutations will usually take place in DNA. And those mutations are usually going to occur during replication in many cases. But it's more catastrophic for the cell to have a mutation in the DNA because this is your original precious copy. Let's say you did have a mutation in one of these bases. Remember, the RNA transcripts are produced and then broken down, produced and broken down, produced and broken down. So you may have a change in one of the bases of RNA, but it's not going to last for that long. You'll have that destroyed pretty quickly. Mutations really have the catastrophic event if it takes place in DNA. So usually when we think of a mutation, we're talking about any change in the sequence of DNA. And then looking at what types of mutations are possible. Let's get these down. And just have my list of possible ones. So we say sometimes you can have a point mutation. Which is just a change, and I'm going to use that delta triangle in a nucleotide. Sometimes you'll hear me refer to that as just a base. So a change in the nucleotide or a base. And sometimes those point mutations can have a few effects as well. So sometimes you'll have a silent mutation. This will be no change in amino acid. Sometimes you can have a missense mutation, where you do have a change in amino acid. Sometimes you can have a nonsense mutation. And this might be where a change in amino acid to a stop. And then the last one will be what's called a frame shift mutation. And this is usually because of the addition or deletion of one or more bases. All right, so I'm going to have a few different examples of those up on the board and ask you to tell me what they mean. Alright, so let's say the original command for the cell was something that we could understand. Took the cat out. An instruction that tells you what to do, written in 3-letter words. And what if you had something like maybe what would you call this type of mutation?
Missense?
Could be almost a missense, but how about a point mutation where you've just changed one base? So I've just changed this E to an H. And a point mutation in this case, it's a little bit of a missense since you have changed this word, but could the cell or you still understand what to do? So probably you could still understand what you're supposed to do. What if you had a change where it was so maybe you've changed a whole codon, so now the amino acid is really put the dog out. This might change the command for you. So this would be that missense mutation. Here's your point mutation. And how about something like put? You'd probably say that looks like nonsense, because you don't have any idea. Nonsense mutations always shorten the command or shorten the protein. Right. And so your nonsense is shortened quite a bit. And you can tell that that's not going to give you an idea of what you're really supposed to do. Lots of things you could put in many places. And then something like put heck auto oot. That might not be something you can understand. and you might spend hours trying to figure it out. But you'll notice it's going to be considered your frame shift mutation. And how did it shift the reading frame? What was it? What kind of mutation happened? So it's just the deletion of a T. But you delete one letter and shift the reading frame, and now you can see the rest of the message gets thrown out of whack. So adding or deleting one or more bases can be really catastrophic for an instruction in a cell. When you start to think about these as proteins produced in a cell, not producing a protein can have a catastrophic effect if the protein's needed. But what about producing a totally faulty protein? Could have haywire types of consequences through the cell. So this can be almost as devastating for a cell as well, versus these, the cell could get by probably. Very mild types of mutations. Alright, so that's all we'll think about for those types of mutations for now.
So the one that has the word put was a known set, you said?
So put without anything means the message has stopped too early. And whenever it stops too early, you know a stop codon has been added.